Hello everyone. Today we will be mainly learning the chapter of liver biochemistry, as you can see here. So we'll mainly discuss about what are the metabolic processes that takes place in the liver and the role of liver as a major organ of metabolism. I'm pretty sure you all by now know what are the functions of liver basically. What are the important functions? So just to have a review, let's check out what are the main functions of liver that we have. Now, liver is considered as the major organ that helps us to metabolize nutrients. For example, glucose, lipids, proteins. These metabolisms, for metabolism of these nutritional substances, liver plays a very major role. Also, liver is known to produce plasma proteins, which we have already finished the chapter of plasma proteins. And we have seen that the plasma proteins have so many important functions. Liver also plays roles in the production of lipoproteins and cholesterol as well. From the previous chapter, you have also learned that liver plays also in the urea cycle. So it plays a very important role also in nitrogen metabolism. Liver also plays roles in the production of bile acids. Now, bile have several functions that we'll go through in today's lecture, and we'll see how bile helps us in several manner. But keep in mind that biles are also synthesized in the liver. Some people make mistakes and think that biles are synthesized in the gallbladder. No, gallbladder is a location where bile is stored basically, not synthesized. Lastly, liver also plays very important role in detoxification process. Okay, in detoxification process, what does liver do? Liver basically will detoxify toxin substances. Example, let's take medicines, hormones, like this. Liver is also known to produce the vitamins and such as A, D, E, B. These are fat soluble vitamins. These vitamins are known to be produced by liver. Basically, liver plays a major role in the metabolism. Okay, mainly you'll see that liver plays roles in the uptake of nutrients from the intestine via the portal vein. What nutrients? Basically glucose, fatty acids, then we have amino acids. These are all absorbed from the GI tract via the portal vein. So after uptake, liver plays roles in converting these nutrients into intermediate products. Liver also plays roles in storage. For example, it can store glycogen, it can store iron and vitamins. Liver is known as a source to supply certain essential compounds as well. For example, when we do gluconeogenesis, synthesis of new glucose, glycogen can be converted into glucose, which is stored in the liver. Okay, liver playing role in the metabolism of nitrogen substances, detoxification, also in secretion of cholesterol, bilirubin, and so on. So let's first start one by one and check what role is the liver playing in all of these. So let's first begins with our carbohydrate metabolism. What role does the liver plays in carbohydrate metabolism? So basically, what happens when the nutrients get metabolized, gets digested and ultimately get metabolized, then it ultimately gets converted into an intermediate called glucose 6-phosphate. Okay, how does it happen? The liver will absorb these nutrients from the intestine, and in the intestine, mainly the glucose that we have, it will be phosphorylated. And this phosphorylation process is catalyzed by two enzymes called hexokinase and glucokinase. And as the catalysis takes place, it converts the glucose into glucose 6-phosphatase. We have already learned from carbohydrate metabolism about glucose 6-phosphate. Now, this glucose 6-phosphate can be converted into several other compounds based on the demand of the body. Now, if you see the following diagram, you can have a much more broader understanding about glucose 6-phosphate. So we know that in the process of glycolysis, at one point glucose is converted into glucose 6-phosphate. and ultimately the glucose 6-phosphate then can be converted into pyruvate, which can be then used in the Krebs cycle or citric acid cycle. Similarly, okay, when the glucose 6-phosphate is converted into pyruvate by the process of glycolysis, okay, at the same time when the pyruvate is there and when we need glucose, let's say we are fasting, we are in a state where we have a low concentration of glucose, at that time the process of gluconeogenesis can convert the pyruvate into glucose if required. So this is one way that glucose is being metabolized. Secondly, the glucose 6-phosphate, if we have sufficient amount of glucose in the body, in the state when we are well fed, we are in resting stage, that this glucose 6-phosphate can be converted into glycogen and stored because we have sufficient amount of glucose. But again, if we need glucose, this glycogen can be broken down by the process of glycogenolysis to give us glucose again. Similarly, glucose 6-phosphate can enter another pathway called pentose phosphate pathways. Now these pathways are mainly concerned with our nucleic acid synthesis and lipid synthesis. So the NADPH are being provided through this pathway for nucleic acid synthesis, pentose, etc. and so on. These are basically the fate that we have for our glucose. So in these whole processes, the role of the liver is basically to store the glycogen. That is, store the glycogen that has been synthesized from glucose 6-phosphate to store it. So usually our liver can store up to, let's say, 70 to 100 milligram of glycogen. Glucose 6-phosphate that gets catalyzed from to glucose 6-phosphate is the enzyme can dephosphorylize the glucose 6-phosphate. If we need glucose, then the enzyme glucose 6-phosphatase can act on glucose 6-phosphate, dephosphorylize it and release glucose. But if we have excess of glucose 6-phosphate, in those cases glycogen will be synthesized, and if those are not used, even after forming glycogen, if we have still excess of glucose 6-phosphate, then those will be converted into glucose. Ultimately what happens, the glucose 6-phosphate gets decomposed to water and carbon dioxide. At the same time, it produces a little amount of energy which are given to the liver cells. Some of the glucose 6-phosphate, as you have seen already in the chart, will be oxidized in the pentophosphate cycle there. So basically our hepatocytes or liver cells have all the mechanisms or all the materials that are required to perform the process of gluconeogenesis, the synthesis of new glucose. So in the liver, glucose can be synthesized from any product or any precursor like lactate, pyruvate, amino acid and so on. So basically what will happen when we are working very much, when our body have a high demand of glucose, at that time the gluconeogenesis, the production of glucose will be switched on, and the lactate that is formed in the muscles will be ultimately transported to liver so that new glucose can be formed and then transported back to the muscles. So liver is playing a very, very important role in the carbohydrate metabolism. Secondly, what role does the liver plays in case of lipid metabolism? So all the process of lipid metabolism basically takes place in the liver. The most important being lipogenesis, that is synthesis of fatty acids and lipids. So for this process to take place, we need the precursor molecule, that is acetyl coenzyme, which is actually formed from glucose and amino acids. Liver being an active organ, what it does is that it will synthesize saturated and monounsaturated fatty acid, which then can be used to synthesize lipids, phospholipids and so on. So liver actually plays a very, very important and central role in the synthesis of cholesterol. 80% of the cholesterol are synthesized here in the liver. And the production of cholesterol is regulated by a mechanism of negative feedback. When we have a sufficient amount of cholesterol in the meal, the liver will decrease its production. And when we have a deficiency, it can increase it. So liver plays a very, very important role. Liver also plays roles in the ketone body synthesis. After the fatty acid are oxidized, there is production of ketone bodies. And liver, what it does, it can transport these ketone bodies to other tissues. For example, to the heart, to muscles, to kidneys, brains, etc. So let's check out this diagram for a better understanding. You can see here, at first, from the intestine, when glucose are absorbed, or from fat cells where we have fatty acids, they can get converted into acetyl coenzyme. In the liver, the acetyl coenzyme can be converted into ketone bodies, which then can be supplied to other muscles and tissues, or can be converted into fatty acids. The fatty acids can play roles in the production of fats, phospholipids, and also it can help in production of certain kinds of fats and cholesterol. The acetyl coenzyme can directly also synthesize cholesterols. Cholesterols plays again role in the production of bile acids, which is the contents of bile, and so on. So, okay, the next is, what role does the liver plays in protein metabolism? Our liver has all the components or the enzyme that is required for the metabolism of amino acid basically. So the liver can use the amino acid in the following ways. Number one, it can perform protein synthesis, or it can decompose the amino acid into its final product. It can transform the amino acid into carbohydrate and lipid. It can make interaction of between amino acids. It can transform the amino acid into different amino acid group and also release it to the blood and transport it to the other organs and tissues which require it. So from the plasma proteins that we have learned, you'll see that 100% of our albumin, the plasma proteins, are made up in liver. 90% of alpha globulins, 50% of beta globulins, okay, clotting factors, fibrinogen, these all are made in the liver. So that's why when we have, let's say, a patient who is showing signs and symptoms of, let's say, bleeding, they have problem with bleeding tendency, gum bleeding, and so on. In that cases, we sometimes do liver function tests to determine the amount of plasma proteins to see if there is a deficiency or not. The liver is also known to produce non-essential amino acid. I hope you guys remember what is non-essential. Those that can be made in the body, right? So liver is also known to produce or synthesize nucleotides, purine and pyrimidine. It can produce or play a role in heme, creatine, etc. So ultimately, what does the liver do for protein metabolism? Number one, it plays roles in deamination of amino acids. If we have excess of it, it can play roles in protein synthesis. For example, it can produce plasma proteins, clotting factors. It can uptake ammonia and play a role in urea synthesis. It plays roles in the catabolism of purines to form uric acids. It can also uptake bilirubin, conjugate them, and excrete them. So liver plays a very, very important role in bilirubin metabolism, which we will be learning later on this class. Next thing that the liver does importantly is biotransformation of xenobiotics, that is detoxification of drugs, toxin, etc. Liver also plays roles in the metabolism of vitamins. Okay, for example, it does a process or mechanism called hydroxylation of calciols to calcidiols. Plays roles in iron and copper metabolism as well. So now let's put our focus on how does the liver does the process of detoxification. So before we know about it in detail, we need to know a term called xenobiotics. Please pay attention. What is xenobiotics? So these are basically foreign compounds which enters the body by any means. So this includes basically drugs, carcinogens, compounds that are found in the environment, such as insecticides, herbicides, and somehow they have gained access to the body. There are certain internal xenobiotics as well, that is substances which are produced inside the body and have toxic properties. For example, ammonia, bilirubin, certain hormones, these are needed to be also deactivated or made inactivated. So this whole process takes place in our liver, and we are calling it the detoxification process, and it will be one of the major function of the livers. Okay, now let's check out what basically are the reaction that is involved in this process and how does it happen. See here, the ways by which xenobiotics are biotransformed in the liver. So reaction that basically takes place includes hydrolysis, reduction, and oxidation. So there are two phases of detoxification, phase 1 and phase 2. There are several enzymes that are present. One of the important one being this one, cytochrome P450. Okay, we are coming to that later on. So let's first start with the phases. Phase 1. So basically in phase 1, mainly the process that involves are hydroxylation, which will be carried out by the enzyme known as monooxygenases or cytochrome P450. This enzyme can also do deamination, dehalogenation, desulfuration, epooxidation, peroxidation, and so on. So basically these hydrolysis reaction and non-P450 catalysis reaction will occur in the phase 2. But other than that, hydrolysis, reduction, oxidation, these basically will take place in phase 1 of detoxification. So basically if you see this diagram, see here, the drugs that we have or certain hormones, certain chemical substances, okay, which are toxic to the body, they will be transformed via phase 1 reaction into some intermediate compounds. Phase 1 reaction can be of several kinds, which we have already seen. For example, if you see the drug acetyl salicylic acid, which is mainly found in aspirin and so on. So if we do hydrolysis of this drug, we may convert them into salicylate in phase 1. Similarly, certain chemicals such as non-epinephrine can be converted into methyl norepinephrine. Now, during phase 1, the major role is played by our cytochrome P450. These are a group of enzymes, and their higher concentration are found in endoplasmic reticulum of the liver cells. They will mainly do catalysis of oxygen atom into substrates, and also will do oxidation. Electrons are transferred from NADPH to cytochrome P40 through the help of flavoprotein. Now once phase 1 is done, the liver then begins the process of phase 2. During phase 2, what will happen? The products that we have obtained from phase 1, that is the hydroxylated or other compound that is produced in phase 1, now will be converted by specific enzyme into various polar metabolites. How? By the process of conjugation. With whom? With glucuronic acid, sulfate, acetate, and so on. So please, in many questions in licensing exams, also during your MBBS, this is a common question that is asked. In which phase of detoxification the process of conjugation takes place? Do remember that conjugation will only take place in phase 2. In phase 1, what we are getting? Mainly hydrolysis, oxidation, reduction. But conjugation will always take place in phase 2. So in certain cases, what happens during phase 1, some of the xenobiotics may be converted and may be made inactive. In these circumstances, the original xenobiotics that becomes inactive are referred as prodrugs or procarcinogens. In other cases, during phase 1 reaction, active compounds can be converted into less active or inactive forms before the process of conjugation. During conjugation, what will happen basically, they will convert the active products of the phase 1 to less active or inactive species. So the main purpose of phase 2 is this, to make them less active, less toxic, and so on. Ultimately, these less toxic and less active substances will be eliminated from the body via urine or via bile. Okay, so this is our phase 2 reaction here. All the products that we have gotten from phase 1 will now go through conjugation, will be made inactive, less toxic, water soluble, and then excreted from the body. So during phase 2, at least 5 types of reaction takes place. This includes number 1, glucuronation. UDP-glucuronic acid will donate glucuronide, and the enzyme gluconeotransferases that is present in endoplasmic reticulum and cytoplasm will do the catalysis. For example, bilirubin, thyroxine, these products can get attached with glucuronal UDP glucuronic acid and may become less toxic, less active. Second reaction that takes place is called sulfation. For example, alcohols, phenols, they become sulfated and may become less active. The tripeptide glucuron is mainly consists of glutamic acid, cysteine, and glycine. Also, we abbreviate it as GSH. You have already learned in the protein metabolism chapter. The glucuron can help us to make a number of xenobiotics deactivated or inactivated. So as those xenobiotics get conjugated with glucuron, they may become less active. Number 4, acetylation. So basically, acetyl coenzyme may react with certain xenobiotics, and the enzyme acetyltransferase that is found in the cytoplasm of several tissues, specifically liver, may help in performing this reaction. So certain drugs, okay, such as isoniazid used for tuberculosis, can be made deactivated or less toxic. Number 5, the last one, is methylation. Certain xenobiotics can, such as amines, phenol, these compounds may get methylated and may become less toxic as well. So basically, these are the 5 reactions that takes place in the phase 2. So do remember about these 5 reactions. Now that we have learned about the role of liver in the metabolism of carbohydrate, protein, vitamins, etc.,